Hey dear students, welcome to my YouTube channel. So in today's video we will study about the electrophoresis of nucleic acid. Firstly we will study the DNA, next RNA. So for the DNA we used various techniques. Firstly we used mostly agarose gel electrophoresis. After that we will study the DNA sequencing and pulse field methods, but mostly we use the agarose gel electrophoresis. Why? We all know that the DNA molecules and their fragments are larger in size as comparison to protein. Since we all know that for the protein analysis by the 2D gel electrophoresis, we use the paste technique, that is the polyacrylamide gel. Okay, so this is the largest size of DNA molecules and their fragments cannot cross the paste technique. So that is why the larger size of agarose gel is required. For example, we have the plasmid PBr 322. That size is MR 2.4 into 10 raised to power 6. So if we talk about in base pair, so it is the about the 4.3 kV. Okay, so when taking about a now if we now talk about the single stranded DNA, next we will study the double stranded DNA. Okay, so single stranded DNA is common to differ size in terms of nucleotides, since we all know that the charge of DNA fragments is all same, so we cannot we cannot separate it on the basis of charge. Okay, so DNA is the same charge, so all the DNA will move on the anode same speed. Okay, same speed, so there is a difficulty to separate because they have the same charge. However, separation in the agarose gel achieved because the resistance to their movement caused by the gel matrix. Slightly we can identify on the basis of the resistant to gel matrix, but mostly we avoid on the basis of charge because they carried the same charge. So that is why the larger molecules we identify by the size. Okay, on the basis of size, smallest molecules move faster. This is analogous to the separation of proteins in the STS page. This is the same in proteins. We also study it based on the smaller size will move faster and larger size move slower. Okay, this is analogous to protein sample. So now if we talk about the double stranded DNA molecules are from the relatively stiff rod, longer molecule endon and passing to the pore, the DNA molecule will experience the drag. So the longer molecules retire lowered by the each pore, and the small double-stranded DNA molecules show the sideways movement. That is, the sideways movements will be more flexible single-stranded DNA. So now if we talk about the gel amount, so gel containing about the 0.3% agarose gel will separate the double-stranded DNA molecules between 5 to 60 kV. As the gel amount decrease, the kV increase. Now, if we increase the gel, 2% gels, agarose gel, the samples can be separate between the 0.1 and 3 kV. But mostly, optimum 0.8% gel size suitable for separating the DNA molecules in between the 0.5 to 10 kV. Okay, so we all know that agarose gel separate the DNA on the basis of their size. Okay, the MR, this is size of DNA fragment, may be determined from its electrophoretic mobility. How they faster or slower? So by running a number of standard DNA markers of known MR on the same gel, this is mostly achieved by running sample on the bacteriophage lambda DNA. That size is 49 kb. So that cleave bacteriophage lambda DNA that has cleaved with the restriction enzymes such as the EcoRI. Since the base sequence of lambda DNA is known, 49 kb, and the cleavage sites of EcoRI are known, so this generates the fragments of accurately known size. Okay, so now if we talk about the gels preparation, so this is the preparation of gel. So DNA gels are invariably horizontal, submarine, or we can say submerged gels. So agarose gel exposed, this is a Agarose gel we are boiling here, okay? So agarose gel are buffered by boiling. This is a agarose gel we buffered by boiling. So after that we poured into the glass. एक minute. So here we are boiling the agarose gel. Firstly we dissolve the agarose gel dissolved in buffer by boiling, and then after that it poured onto the glass, okay? So this glass was surrounded by a adhesive tape. This is a adhesive tape, okay? So we can adhesive tape use here, okay? So after that we loading the plastic well forming template. This is a plastic well forming. This is a well, this is a tape, adhesive tape. This is a well. I am just here drawing for the understanding, okay? Not write in your exams. So we here plastic well forming a template. I repeat, firstly agarose gel dissolved in gel buffer by boiling, okay? So it poured into the glass, so surrounded by a wall of adhesive tape, okay, glass. So after that loading the plastic well forming a template, okay? So in the poured solution the gel is placed, this gel is placed in electrophoresis, okay? This gel is placed in electrophoresis tank and covered with the buffer. This is the open, so this is the covered by the buffer. So and the sample loaded by, and this I am preparing here sample, okay? And this sample is directly injecting into the bell here, okay? This is a sample I injecting here into the well. Now how we can prepare sample, okay? Sample are prepared by the dissolving in the buffer solution that contain the sucrose, glycerol and ficol. Why we use sucrose, glycerol and ficol? Because this make the sample solution dense and allows it to sink to bottom, okay? So we can use also dyes such as bromophenol blue, which gives the which is the phenolphthalic anionic dye, and it acts with the basic dyes mostly, and this gives the blue color, okay? We also study in histology and histochemistry. So we use here dye bromophenol blue in the sample, so it make easier to see the sample for the why the fluorescence, okay, is being loaded, and also act as a marker of the electrophoresis friend. So we don't use here stacking gel. Now stacking gel, what is? So it increase the mobility, okay? No stacking gel is needed because of the mobility of DNA is already greater in the well than in the gel. In the well, the mobility of DNA is greater as comparison to gel, okay? So if we talk about general purpose, so so it should be 25 centimeter long and 12 centimeter wide, and run with the voltage gradient of 1.5 voltage per centimeter overnight. For rapid analysis that do not need extensive separation of DNA molecule, it can be used mini gels, okay? That use a distance of 10 centimeter long, okay? So in this way we can obtain the result in 2-3 hours. So now if we talk about the dyes, so once the system has done, the DNA gels need to be stained with some dyes like ethidium bromide and the SIVR green, okay? So the gel is rinsed in a solution of the fluorescence dye and viewed under the UV light. So alternately dye can be dissolved in the liquefied agarose gel just before the pouring the gel. So dyes such as the ethidium bromide and the SIVR green uh, we use here that bind with the stack base pair of DNA, okay? That bind with the stack base pair of DNA. The dye concentration therefore build up the site of DNA band. So this dye concentrations build the site of DNA bands. We can see here bands. So we can visualize it, Okay, for example, if we talk about the thetium bromide, so it gives the orange red color, and SiVr green, as the name suggests, as the name represents the green color. Okay, so now some is the precautions. So it is essential to protect our eyes with googles, and if there is a long time work with the ultraviolet, so we can use the plastic bag that covers the whole face, so that we can protect ourselves from the ultraviolet light. Okay, so this is the all about the agrostia lactophoresis. This is the photograph showing the tracks from the, this is for the, you can see here, 6.14. Okay, so this is an experiment done for this bacteriophage lambda DNA. Okay, next we have DNA sequencing gel. I already told that the agarose gel used mostly as comparison to other method. Okay, so but still this DNA sequencing gels for the such as the DNA footprinting. Okay, so we use the DNA sequencing gel in place of di-deoxy sequencing for the routine applications. Okay, this is replaced by di-deoxy sequencing for the routine application. But in some case like DNA footprinting, we use this technique. So we all know that the gross gel electrophoresis of DNA is highly suitable for the short DNA molecules as well as long and large size molecules. The final analysis involved the separating the single standard DNA molecule shorter than the this size and differing size is 1 nt. To achieve this, it is necessary to have small pore gel in the DNA sequencing gel. Okay, so we acrylamide gels are used instead of agarose. Okay, so for example, if we have 3.5% polyacrylamide gels are used to separate the DNA in the race this. So 12% gels to dissolve the fragment of between the 20 to 100 nt. So this is some examples, like we can use this for the separation of small pore size DNA. So this is other examples. Okay, this is not as much importance as comparison to agarose gel. Next we have pulse field electrophoresis. Okay, the agarose gel method for DNA described above, we already discussed about it. So the method basically involves, this method pulse field gel electrophoresis involves the electrophoresis in agarose where two electric field are applied. Okay, in this we use pulse field electrophoresis, we use the two electric field alternately at different angles for defined time periods. We, for example, 60 seconds. So activation of the first electric field causes the coiled molecule. If we use the first electric field, it will cause the coiled. Okay, so if we use the first electric field, here we'll use the two, but if we use first electric field, that causes the coiled molecule to be stretched in the horizontal plane. Okay, and start to move through the gel, and interruption of this field, the application of second field force molecule to move into new direction, the second field move in the new direction. So this length dependent relaxation behavior to move into the new direction owing to a length dependent relaxation behavior when a long chain molecule undergoes a change in the electric field. So by the electric field, that is the two electric field, we can identify the DNA molecules on the basis of their how they distance invaried. Okay, so the smaller molecules quicker it lies itself with the new field. We all know that if we have small molecules, they will move faster as comparison to large molecules. Okay, so we can identify the size on the basis of size by using the pulse field gel electrophoresis. So this is the auto radiograph sequencing gel samples were prepared using the Sanger dideoxy method of DNA sequencing. So this is a DNA sequencing method. So we studied here two electric fields. Okay, so in this way, with continual reversing of field, one is moving this direction, other is moving in another direction, second electric field moving another direction. So the small molecule had larger molecules and separate according to the size. So pulse field gel electrophoresis proved that the particular used to identify the cause of outbreak of bacterial we use in bacterial foodborne illness, for example the salmonella infections like typhoid fever, so we can use in this. Okay, so next we have example. For example, salmonella species when ingested with the restriction enzyme, that is the XVA1, so it gives around the 15 fragment ranging from the 25 kb to 680 kb. So we all know that salmonella causes typhoid, which is caused by the contaminated water and food consumption. So we are analyzing here the salmonella typhi, okay, that have the 15 fragment and size is the 25 kb to 680 kb. So the patterns of fragments, or we can say the fingerprint is a unique to that stain. If the same fingerprint is found from the bacteria from other infected people, then it can be assumed that that person was infected by the typhoid fever, or we can say the salmonella bacterium. So thus by comparing their eating habits, food sources, etc., source of infection can be tracked by the restaurant food item. So we all know that if we wanna to avoid the salmonella typhi bacterium, we should protect ourselves, we should protect our hands and feet, and for the better hygiene. Okay, so we can use the we can use the sanitizer for the protection. So this is the all about the gel electrophoresis of nucleic acid. Firstly we done about the DNA. Okay, next we are studying about RNA. This is a very short, okay, short time. Okay, so electrophoresis of RNA, like that of DNA, electrophoresis of RNA is usually carried out in agarose gel. Okay, and the principle of separation based on size. Principle of separation is based on size. We also identify RNA on the basis of size. Same, almost. Okay, so one requires a rapid method for checking the integrity of RNAs immediately following the extraction and before the deciding whether to process this whether. So this can be achieved by the electrophoresis in 2% gel in about 1 hour. So we use here agarose gel about the 1 hour. For example, ribosomal RNA for 18S and 20S are clearly resolved by any degradation, or DNA contamination is seen easily. So if we use this, so we can easily analyze the contamination of any fragments, any protein fragment, or we can say DNA contamination can easily visualize. So this can achieved on a acrylamide gradient gel that is approximate 2.5 to 5% with the overnight run. Okay, 1 day. So both this method, this and this, so both these methods involving the running the native RNA, so there will be almost certainly be secondary structure within the RNA molecule owing to the intramolecular hydrogen bonding. So there will be forming the intramolecular that have the same molecular hydrogen bonding. For example, is a clover leaf, we will see in the later on. Okay, in the structure 4.6. So for this reason, native DNA run on the gel. Okay, this native DNA run on the gel can be stained and visualized with the ethidium bromide. Similar to DNA, in DNA electrophoresis, we also use this dye. Okay, ethidium bromide. Here we use the ethidium bromide. This, okay, that gives the color orange red. Okay, similarly, we use here the ethidium bromide dye. So however, if the study of objective to determine the RNA size by gel electrophoresis, then full denaturation of RNA is needed. Denaturation means the breaking of the hydrogen bond. So if we require the determines the full detail, so we should denaturation of the DNA is needed to prevent the hydrogen bond formation. So we break the hydrogen bond that will otherwise affect the electrophore if there is a presence of hydrogen bond. Okay, so that will affect the electrophoretic mobility. Okay, that cannot easily move. Okay, so that is why we use here denaturation of DNA, that is the breaking of the hydrogen bond. So there are three denaturation agents which used for the denaturation of DNA, that is the formaldehyde, glyoxal and the methyl mercury hydroxide. This is the most important three I repeat: formaldehyde, glyoxal and methyl mercury hydroxide, that are compatibly with both the DNA and agarose. Either one of these may be incorporated in the agarose gel and electrophoresis buffer, and the sample is heat denatured. We should done the heat denaturation, the presence of denaturation prior to the electrophoresis. After heat denaturation, each of these agents from the ducts with the amino groups of the guanine and the uracil of the RNA, thereby preventing the hydrogen bond reformation at the room temperature. Okay, so during the electrophoresis, it is also necessary to run the denaturation gels, like we studied about the formaldehyde, glycol and the methyl mercury hydroxide. So this is a very essential use in the gel RNA gel electrophoresis. Okay, so we can also use north blot techniques and probe to ensure that the base sequence is available to the probe. You can see the structure. This is a separation of yeast RNA by using the 1.5% agarose gel for the RNA analysis. So denatured RNA stain only very weakly with the ethidium bromide. So we can also use here Recridine orange is commonly used to visualize the RNA on the denaturing gel. If we use here denaturation, any denatured materials like glyoxal, so we can use here Icrine orange as comparison to ethidium bromide stain. Okay, so however it should be noted that many workers will be using the radio levels RNA accordingly identify the bands of autoradiography. So this is the examples of the gel electrophoresis of RNA which is used in the yeast RNA. So this is the all about the electrophoresis of DNA and RNA. I hope this video will helpful for you, and this was done by Wilson and Walkers. Okay, so if you have any query you can simply comment in comment section, and if you need another topic of the Wilson and Walker work, so you can comment. I will upload soon. So that's all about today's video. Thank you.